In this EReport, we will cover the formation and structure of cytoskeletons fibers, cell crawling, G-protein coupled receptors, cells progression through the cell cycle with cyclins and CDKs, cell junctions, and cancer. Intermediate filaments are made of pairs of proteins that come together into a twisted strand. These twisted strands are called dimers. These then come together in two to create a tetramer. 8 of them come together to make a rope-like structure. Please watch the video to see how it is done. Next are microtubules. They are made from tubulin, which are globular proteins that stack themselves. You can see the alpha and beta tubulins there. They come together like a tube-like structure, and they disassemble and reassemble quickly, starting from the centrosome and ending at the cell membrane. If they do not hit their target, then they are disassembled. Actin is the next filament. It is helpful for the cell to move, and it also helps it keep its shape. Actin are thin light rope structures that are created by 2 sets of proteins that wrap themselves together. It is the smallest, but also one of. Next is cell crawling. In order for that to happen, the actin must bundle up and create a lamellipodia or filopodia and stretch out. Once it is stretched out, integrins will lock into extracellular surfaces. Once they are stuck and have a firm spot, then the rest of the cell must follow into the anchor points with myosin and actin movement. G-protein coupled receptors recept a singling molecule, and from there, the G-protein, made out of an alpha, beta, and gamma, will react to an enzyme or a channel. These channels then will send out second messengers, depending on what channel or enzyme is started. The first second messenger is cAMP. cAMP is when the gamma from the G-protein comes over to adenylate cyclase, an enzyme that turns ATP into cAMP. The cAMP is a molecule that will bring the message from outside the cell inside the cell and activates kinase A for more cellular response. The second one is calcium. The calcium starts when phosphorylase C hydrolyzes into PIP2, which gets split into IP3 and DAG. IP3 goes to the UR to release calcium, which will combine when DAG activates kinase C and will bind together and make them both more active. The cell progresses through the cell cycle with only green go from checkpoints. These checkpoints are regulated by kinase, or cyclin-dependent kinases to be. Here is a CDK inhibitor, P53. When there's DNA damage, P53 is phosphorylated and active. When it is active, it makes P21. P21 then becomes more like a bear hugger and hugs onto the cyclin D, CDK, allowing it not to move on to the next checkpoint. Next is cell junction, starting with the tight junction. The tight junction happens between 2 epithelial cells. They come together, alternating claudin and O-claudin intermembrane proteins. These create a locked seal that molecules that it cannot go through, including on the skin and intestines where you don't want molecules to be going through. Tight junctions are also good for defining the membrane domain because no molecule will get through there, and therefore it will. Next, there's adherent junctions. These connect 2 epithelial cells between cadherins. Cadherins are transmembrane proteins that connect. Desmosomes are another type of cell junction. They are when proteins within the cell connect to proteins that are within the membrane, and they combine 2 epithelial cells together through the actin filaments within the cells. Next are the hemiadesmosomes. They bind the epithelial cells to the basal lamina. This happens through proteins connecting along. Next are gap junctions. Gap junctions are like little tunnels between 2 Cancer cells are like normal cells, except they no longer follow the rules. They grow with no limitation and are selfish to the organism. Unlike other cells that conform and do as they are supposed to, cancer